The NEET Aspirants 2026, welcome to the evening batch. So yes, in our previous class, what all we dealt? We have dealt with the major terms which we are dealing in the respiration, fine, respiratory substrate, cellular respiration, and then we have also dealt why ATP is called as cellular energy, and then we have looked into do plants breathe, what are the things which are present in the plant through which plant breathes. Is there any complex respiratory system in the plant? No, but then it has what? Stomata and lenticels, fine. And then after that we have looked into glycolysis. Glycolysis, which is the common respiratory pathway both in the aerobic and anaerobic respiration. Right, respiration is of two types based on the presence or absence of oxygen for the oxidation of food materials, and respiration is also of two types based on the respiratory substrate that it is going to get oxidized to. Okay. So, if the respiratory substrate is proteins, then it is called as what? Protoplasmic respiration. If the respiratory substrate is fats or carbohydrates, then it is floating respiration. That classification is given by Blackman. Okay. And then glycolysis we have learned, which is the common pathway both for aerobic and anaerobic respiration. Then we have dealt with anaerobic respiration, both lactic acid fermentation and alcoholic fermentation. Now we are going to look into complete steps, detailed steps of aerobic respiration. First we will deal with oxidative decarboxylation, then Krebs cycle, okay, Krebs cycle, electron transport system, amphibulic pathway, respiratory quotient, and at the end we will be solving the objective as well. Fine, beta, let us get started. So yes, we have discussed that once the glucose is being synthesized in a cell, okay, glucose after getting synthesized in the cell, how glucose is being transported to other cells as sucrose. Okay. So, sucrose entering into a cell, sucrose first gets sliced to what, Nana? Glucose and fructose. By what enzyme? Invertase. By the enzyme invertase, sucrose is sliced to what? Glucose and fructose. This glucose will start the process of glycolysis in cytoplasm. Fine. At the end of glycolysis, what forms? 2-pyruvic acid. At the end of glycolysis, 2-pyruvic acid will be formed. This 2-pyruvic acid will directly go and land where, beta? Into the mitochondrial matrix. Okay, into the mitochondrial matrix. Yes, into the mitochondrial matrix. Pyruvic acid will directly go and land into the mitochondrial matrix. So, here if you see, mitochondria will have what? Outer mitochondrial membrane, outer mitochondrial membrane, inner mitochondrial membrane which has enfoldings called as what? Cristae, and then this is what? Intermembrane space, and here inside it is mitochondrial matrix. So, this pyruvic acid landing into the mitochondrial matrix, first step that happens is oxidative decarboxylation. Fine, oxidative decarboxylation. After oxidative decarboxylation, TCA cycle or Krebs cycle will also occur where? Inside the mitochondrial matrix. After TCA cycle or Krebs cycle, mitochondrial matrix will be rich with all the energy, NADH, FADH2 and ATP. But then to convert the NADH and FADH2, which is a very energy rich molecule, into the simpler molecules ATP, ETS will be operated in the intermembrane space, in the inner mitochondrial membrane. Fine, ma? So, first we will discuss about oxidative decarboxylation. What is oxidative decarboxylation? What are the substrates for oxidative decarboxylation? Pyruvate. Yes, pyruvate. Pyruvate, which is present in the mitochondrial matrix, will link up with coenzyme A. Okay. So, in one cycle of oxidative decarboxylation I am discussing with, out of 2 pyruvic acid, one has started. Fine. Pyruvic acid, CoA and NAD plus. Fine. In the presence of the enzyme pyruvate dehydrogenase. decarboxylase or dehydrogenase, one and the same, because NAD plus will be dehydrogenated to NADH, right. So, pyruvate dehydrogenase or pyruvate decarboxylase also fine. Both the enzymes, pyruvate dehydrogenase, decarboxylase. Pyruvate dehydrogenase or pyruvate decarboxylase. This enzyme will have a cofactor called as magnesium ions. Okay, pyruvate dehydrogenase, pyruvate decarboxylase, they will be having a cofactor, that is magnesium. Fine. And here acetyl CoA is formed. Acetyl CoA. Pyruvic acid is 3 carbon compound. Acetyl CoA is 2 carbon compound. Acetyl CoA is 2 carbon compound. And this forms out to be NADH plus H plus. Okay, NADH plus H plus carbon dioxide. Okay, so the same thing if you have to consider for 1 glucose. For 1 glucose means 2 pyruvic acids, 2 pyruvates, 2 ODs will happen inside the mitochondrial matrix. As such, for 1 glucose molecule in oxidative decarboxylation, how much of the energy will be released beta? 2 NADH, and how much of carbon dioxide will be released? 2 CO2 will be released. Oxidative decarboxylation is clear to you, right? Yeah. Now here acetyl CoA will start TCA cycle. Okay, acetyl CoA will be starting up the TCA cycle. Look at here carefully. In tricarboxylic acid cycle, TCA cycle, which is also called as Krebs cycle, named after the scientist Hans Krebs, which is named after the scientist Hans Krebs. Fine, where he has elucidated the Krebs cycle from flight muscles of a pigeon. Okay, from flight muscles of pigeon. Fine. So, which molecule is starting up this reaction? Acetyl CoA. Acetyl CoA is how many carbon beta? 2 carbon. It is binding up with oxaloacetic acid, which is 4 carbon. Okay, acetyl CoA, which is 2 carbon, is binding up with oxaloacetate OAA, that is 4 carbon. By the enzyme citrate synthase, it forms out to be what, Nana? Citrate. Citrate is how many carbon? 6 carbon. Fine. So, this 6 carbon will get converted to isocitrate. Isocitrate gets decarboxylated by the enzyme isocitrate dehydrogenase into alpha ketoglutarate acid. Isocitrate is 6 carbon compound, alpha ketoglutarate will be 5 carbon compound, and here decarboxylation happens, right. 1 carbon dioxide will be decarboxylated and 1 NADH is synthesized, 1 NADH. Fine. Now, this alpha ketoglutarate acid again gets decarboxylated to what? Succinyl coenzyme A by the enzyme alpha ketoglutarate dehydrogenase. Fine, right, ma? So, here also 1 NADH will be released out at this step as well. Succinyl coenzyme A to succinate. Okay, succinyl coenzyme A to succinate by the enzyme what? Succinyl CoA synthase. So, here substrate level phosphorylation happens. Fine, because phosphate group is being extracted from this substrate succinyl CoA and being added to what? To form GTP or ATP. Understood? So, SLP happens over here and succinate is again 4 carbon compound will be turned to what? Fumarate by the enzyme succinate dehydrogenase. All the enzymes which are present in the Krebs cycle are soluble inside the mitochondrial matrix, but succinate dehydrogenase is attached to the inner mitochondrial membrane. Do remember that. Now, this substrate is being converted to what? Fumarate, and how much amount of energy is released, ma? 1 FADH2. Fine, and then fumarate to malate, which is 4 carbon, by the enzyme fumarase, malate to oxaloacetic acid by the enzyme malate dehydrogenase, 1 NADH will be released out. Understood? So, in this complete cycle, tell me at what points carbon dioxide is being released? First isocitrate to alpha ketoglutarate to succinyl coenzyme A. If we have to consider for one glucose molecule, at 3 sites in the mitochondrial matrix, carbon dioxide is being released. Yes or no? At 3 sites. 2 CO2 first released where? At the oxidative decarboxylation in the mitochondrial matrix, and from isocitrate to alpha ketoglutarate, 2 carbon dioxide for 1 glucose I am saying, right? From where? Isocitrate to alpha ketoglutarate, fine, and then 2 carbon dioxides will be released again from where? Alpha ketoglutarate to succinyl CoA. Remember this, very important. Fine. So, complete oxidation of glucose happened or not? Happened. Complete oxidation of glucose. Now, one more major point. What are the sites where NADH, FADH2 and GTP are being released in the Krebs cycle? Listen carefully. At 3 points NADH is being released in Krebs cycle. At 3 points NADH is released. What are they? Yes. First is isocitrate to alpha ketoglutarate, and second site is alpha ketoglutarate to succinyl CoA, and last is malate to oxaloacetate or malic acid to oxaloacetic acid, right? At one point FADH2 is released, where? Succinate to fumarate, very good, succinate to fumarate. At one point ATP is released, succinyl CoA to succinate. So, these are the major points and steps where you have to remember. Okay, energetics are very important. Now, mitochondrial matrix is rich with energy, right? In combiningly for one OD and one Krebs cycle, how much amount of energy is being released in mitochondrial matrix? That is the question. Okay. Inside the mitochondrial matrix, for one complete cycle of oxidative decarboxylation and TCA cycle, how much amount of energy is released? 1 NADH plus here 3 NADH plus how much FADH2? 1 FADH2,
1 GTP.
That's it, 1 GTP. Convert everything into ATP. 1 NADH is equal to 3 ATP. So, total here 4 ADP are there. How much ATP beta? 12 ATP. And 1 FADH2 is equal to?
2 ATP.
Yes, 1 FADH2 is equal to 2 ATP. So, on total how much? 2 ATP.
1 GTP is 1 ATP.
Yes. Total how much ATP?
1.
Total 15 ATP. So, one complete cycle of oxidative decarboxylation and TCA cycle will give out 15 ATP in the mitochondrial matrix. Clear, right? Now, for one glucose, what is the energy sheet? Energy sheet for one glucose.
Glycolysis also.
Yes.
36.
Listen first, na. Energy sheet. We have to consider all the 4 processes.
3
First is what? Glycolysis. Second will be oxidative decarboxylation. Third is TCA cycle. Fourth will be electron transport system. Fine. So, here if you see glycolysis, how much energy is being released, ma? 2 NADH and 2 ATP.
gross.
Okay, gross. And oxidative decarboxylation, 2 ATPs net, gross will be 4, but we have to consider 2 ATP only. Out of 4 ATP, gross ATP, 2 will be utilized by first and third step, right ma? So, we have to take 2 ATP for glycolysis. Oxidative decarboxylation again,
2 NAD
2 NADH.
for whole glucose.
Yes. TCA cycle,
6 NADH,
6 NADH,
2 FADH2,
2 FADH2 and 2 ATP or GTP. Fine. Electron transport system we will look after this, after the complete energy sheet. Now, this we have to convert into ATP, right. So, 2 into 3, how much ATP? 6 ATP, and here 2 ATP, 2 into 2, 4 ATP,
3
sorry, 2 into 3, how much? 6 ATP. Here 6 into 3, how much ma? 18 ATP, fine. And then 2 into 2, 4 ATP, and lastly 2 ATP. Okay. Tell me on total how much ATP is being synthesized or released from one glucose in aerobic respiration? 38 ATP. But then if in your question, if what is the total amount of energy that is being released from aerobic respiration from one glucose, then you will choose 36 ATP, not 38 ATP. Why? It is because from glycolysis the 2 NADH which is released and 2 ATP which is released, this 2 NADH to get into the mitochondrial matrix, a shuttle system will be operated. That shuttle system is called as glyceraldehyde 3-phosphate shuttle system. Okay. It will charge 1 ATP to transport 1 NADH. Okay. 1 NADH to be transported from cytoplasm to mitochondrial matrix. This shuttle system will charge 1 ATP. As such, 2 ATP will be utilized by the shuttle system to transport 2 NADH from cytoplasm to the mitochondrial matrix. Fine. That is the reason in a eukaryotic cell, total amount of energy released in aerobic respiration will be how much? 36 ATP. Any doubt anywhere still? No, right ma? Yeah. So, till here it is clear to us about what? Oxidative decarboxylation and Krebs cycle, right. Here now we will be looking into electron transport system. Electron transport system. Fine. ETS, all the energy rich molecules NADH, FADH2, where are they present? Inside the mitochondrial matrix. Okay. Inside mitochondrial matrix, all the energy rich molecules are present. So, to convert this into ATP, what is operated? 4 complexes are operated in the inner mitochondrial membrane. Electron transport system location is what? Inner mitochondrial membrane or cristae. Fine. So, here complex 1 is what? NADH dehydrogenase. Okay. Complex 2 is FADH2 dehydrogenase or succinate dehydrogenase. Complex 3 is cytochrome BC complex. Complex 4 is cytochrome C oxidase. Okay. So, look at here. What is complex 1? dehydrogenase. Complex 2 is
succinate dehydrogenase.
succinate dehydrogenase. Complex 3 is
Cytochrome BC complex.
yes, cytochrome BC complex. Complex 4 is
Cytochrome C oxidase.
cytochrome C oxidase. Fine. So, here if you see complex 1 NADH dehydrogenase, what does this do? It will oxidize NADH to NAD plus H plus electrons. Okay. Protons, 4 protons will be travelled from the mitochondrial matrix directly to the intermembrane space, whereas 2 electrons will be accepted by flavin mononucleotide, that is FMN protein molecule. Okay. And from here it will be accepted by what? Ferrous sulfur molecule. Fine. First is flavin mononucleotide and it is ferrous sulfur complex. From here electrons are being given to what? Ubiquinone, which is a mobile electron carrier. Remember, what is ubiquinone? Mobile electron carrier. Fine. Ubiquinone will be turned to what? Hydroubiquinone, and protons and electrons will be submitted to complex 3 from complex 1. Understood? Now, yes, will be submitted to what? Complex 3. From here you see in complex 2, that is succinate dehydrogenase, which is being turned to succinate, which is the enzyme that is activated from the substrate succinate to fumarate. Here FADH2 is oxidized to FAD plus H plus electrons. Fine. Same the case, protons and electrons will be pumped into the inner mitochondrial membrane, but then this complex is attached to inner mitochondrial membrane. This is not a transmembrane channel. Okay. So, that is the reason it is not transpanning two sides. That is the reason both protons and electrons are accepted by flavin adenine dinucleotide, FAD. Okay. From here electrons are being submitted to what? Again ferrous sulfur complex. Fine. And then both electrons and protons are given to ubiquinone, then which is a mobile electron carrier, and then both are submitted to what? Third complex, cytochrome BC. Fine. Understood? So, cytochrome BC complex, again electrons and protons will be travelled. Protons are submitted to intermembrane space, whereas electrons are given to cytochrome B, then ferrous sulfur complex and cytochrome C. Here again there is one more mobile electron carrier. What is that? Cytochrome C, which is towards the intermembrane space. Fine. Attaching to the inner mitochondrial membrane, this is towards the intermembrane space. This accepts the electrons and submits it to what? Complex 4. In complex 1, 2 and 3 you can observe ferrous sulfur complex, whereas complex 4 will be having copper molecules and cytochrome A, A3. Fine. At the end the electrons will be accepted by, the ultimate electron acceptor is oxygen molecule. Remember that. Okay. What is the ultimate electron acceptor? O2 is the ultimate electron acceptor. Fine. And here 4 protons are being accumulated over here, of which oxygen takes 2 protons, forms out water molecule. The rest 2 protons are being pumped back to the intermembrane space. Understood? So, in this continuous process, proton accumulation you can see in the intermembrane space, whereas in the mitochondrial matrix there is a deficient in protons. Fine. That is the reason there is a proton gradient that is generated in between the intermembrane space and the mitochondrial matrix, wherein protons are being pumped through the ATP synthase. ATP synthase is activated. This is also known as complex 5, where protons will be pumped in one run. 4 protons will be pumped. Okay. So, this ATP synthase has 2 complexes. What is that? F0, F1 complex. F0 will be in the inner mitochondrial membrane, F1 will be towards the mitochondrial matrix. Understood, ma? So, here this will pump how many protons? 4 protons for the formation of 1 ATP. So, here do you see water molecule formation, oxygen and ATP. Done with the process of aerobic respiration. Any doubts anywhere over here?
F0 is inside or F1 is inside?
F1 is inside the inner mitochondrial membrane. Sorry, F0 is present inside the inner mitochondrial membrane. F1 is towards the mitochondrial matrix. Done? You may expect the comparative question of F0, F1 in considering both the organelles chloroplast and mitochondria. In mitochondria, F0, F1 pump, F0 will be in the membrane, F1 is towards the mitochondrial matrix. In chloroplast, F0 is inside the thylakoid membrane and F1 is towards the stroma. Okay, F1 is towards the stroma. Understood? Any doubts anywhere? Yeah. Next. Amphibolic pathway. What is amphibolic pathway? So, you know, of course, glucose or carbohydrates is the most favored substrate for the respiration. But then we also have other respiratory substrates like what? Fats and proteins. Fine. Fats will be first oxidized to fatty acids and glycerol, where fatty acid will join the cycle as acetyl-CoA. Fine. And glycerol will be joining the cycle as dihydroxyacetone phosphate or phosphoglyceraldehyde. Fine. Now, proteins will first get deaminated to amino acids by the enzymes proteases. Fine. And this amino acids will be joining the cycle at pyruvic acid. So, all the respiratory intermediates that you see in the complete process of aerobic respiration, like you know acetyl-CoA, succinyl-CoA, alpha ketoglutaric acid, all these respiratory substrates or intermediates are the starters of some other biosynthetic pathways. Fine. For example, acetyl-CoA is the raw material for the synthesis of carotenoids, terpenes and gibberellins. Fine, like that. Succinyl-CoA is also, succinyl-CoA is the raw material for the synthesis of carotenoids and cytochrome. Fine. Alpha ketoglutaric acid is the raw material for the synthesis of alkaloids and pyrimidines. Got it? So, what is amphibolic pathway is which includes both anabolic and catabolic processes. Fine. Of course, respiration is what? Catabolic process where the complex molecules are being break to the simpler ones. But at the same time, the respiratory intermediates whichever are present, they are the raw material for the synthesis of certain molecules. Okay, that is why it is called as what? Respiration is called as amphibolic pathway. Done, right? Any doubts anywhere with amphibolic pathway? Here what you have to remember is each respiratory substrate where they are joining the cycle, okay, and Krebs cycle is the common respiratory pathway for all the respiratory substrates. Fine, right? Yeah. Now, the last segment of this chapter, that is respiratory quotient. Respiratory quotient. What is respiratory quotient? Nothing but the ratio between the volume of carbon dioxide evolved by the volume of oxygen consumed. That is the respiratory quotient. So, here if you see, okay, glucose, if it is the respiratory substrates, 6 carbon dioxides are evolved and 6 oxygen is consumed. The RQ value will be 1. If you take fats, for example, tripalmitin, carbon dioxide evolved is 102 carbon dioxide, whereas oxygen consumed is 145. Okay, what will be the RQ value? 0.7. Okay, for proteins if you take ma, just a second. For proteins, it will be what?
CO2 list is
More than the oxygen. So the value will be 0.9. What if the organic acids are the respiratory substrates?
CO2 is released
More carbon dioxide will be released. Then the value will be more than 1. For example, if it is malic acid, the value will be 1.33. Fine. If it is oxalic acid, the value will be 1.4, like that. It will be more than 1. For proteins it will be less than 1 only.
Oxygen is consumed more.
Yes, more oxygen is consumed for oxidation, right, but not as much for fats. And now the question to you, what will be, Sarika, you can ask your doubt ma. What will be the RQ value for anaerobic respiration? Especially for alcoholic fermentation in specific for alcoholic fermentation. Huh?
2 oxygens, 2 CO2 is released, but 0 oxygen. 2 by 0. 2. Infinite. Max
Ain't stronger. 2 by 0 is what? Infinite. Fine. Any doubts anywhere? Next. See, recognize the figure and find out the correct matching. You have to answer this question. Labelings are given and substrates are given. You have, energetics are given. You have to identify the substrates. Where is A over here? A, B, C, D and E. A number of carbons are also given. Identify and say. Both online and offline can answer ma.
options.
Huh?
Option C.
Option C you are saying. Okay. Raghav, what's your option beta? Don't know. Very good. So look here. C is what? Malic acid. Yes, C is malic acid, which is being synthesizing what? Oxaloacetic acid, right?
This is C.
Yeah, C is what? Malic acid. It will be synthesizing oxaloacetic acid. So E is oxaloacetic acid. Good. D is what? Alpha ketoglutaric acid. Fine. And A will be succinic acid. And B is what? Citric acid. Good. Okay, so option C will be the right answer. Next question. In TCA cycle, the formation of isocitrate is followed by 2 successive steps of decarboxylation. What are the products formed after each decarboxylation? Okay. So, isocitrate to
Alpha ketoglutarate.
Yeah. By the enzyme?
Isocitrate. Isocitrate synthase.
Isocitrate dehydrogenase. By the enzyme isocitrate dehydrogenase. Okay.
To succinylCoA.
By the enzyme?
Alpha ketoglutarate dehydrogenase.
Good.
With help of cofactor A.
Good. Okay. So, here both the places carbon dioxide will be released and 2 NADH will be released out. Done? Yeah. So, what will be the right answer now? Option A. Next, this one. All enzymes of TCA cycle are located in mitochondrial matrix except one which is located in inner mitochondrial membrane in eukaryotes and in cytosol in prokaryotes. The enzyme is. Correctly answered. Look at it once again. Raghav, why are you not looking into the question at least? All enzymes of TCA cycle are located in the mitochondrial matrix. All enzymes are soluble except one which is located in the inner mitochondrial membrane in eukaryotes and in cytosol in prokaryotes, and the enzyme is what? Succinate dehydrogenase, fine, which is present in the inner mitochondrial membrane. Next, ETS. You have to label it properly. A, B, C, D and E. Raghav, you can answer beta. Look at it. Good, Manoj? B. Okay. A is what? Mitochondrial matrix. B is intermembrane space and C is cytochrome B because it is cytochrome BC complex, and E will be what? Cytochrome E. E is what? Cytochrome C. It is a mobile electron carrier in between complex 3 and complex 4. This is cytochrome C. And then D is what? Cytochrome B. Yes, option C will be the right answer. Option B will be the right answer. Sorry, B is the right answer. Next question. Take 2 minutes time, look into the question carefully and then answer. Raghav, can you? Okay, no issues. Study the following statements regarding chemiosmotic hypothesis in mitochondria and select the correct ones. F1 headpiece contains the site of synthesis of ATP from ADP plus inorganic phosphate. Fine. So, ATP formation occurs inside the mitochondrial matrix. Option 1 is right. Next 2. F0 part forms the channel through which the protons cross the inner membrane. True. Option 3. For each ATP produced, 2 H plus pass through F0 from the intermembrane space to mitochondrial matrix. No, it is 4 H plus. Option A is the right answer. 1 and 2. And then, okay. So, with this we have exercised the questions as well. With this, the respiration and plants chapter will be done. In our next session, we will be starting plant growth and development. Okay. Thank you.